everyone. So this is an interesting concept that we are going to discuss in this video. So the very basic entity of a biotic component, that is biotic component means a living organism, is that we are all made up of cell, the basic unit cell. So whereas non-living organisms are not made up of cells, whereas living organisms are made up of cells. So we are very clear about it. So each and every cell in turn has got either a poorly developed or well developed nucleus. So in case of bacteria prokaryote, we happen to see a poorly developed nucleus, whereas in eukaryote we see a well developed nucleus. So nucleus in turn has what? And then you all will answer it is going to have DNA. Some will say it as DNA. Some will answer it as chromosome. Some will answer it as gene. So this video is actually meant to have a clear-cut distinction between DNA, genes, chromosomes. Okay, so they are not going to be same. Okay, there's going to be a very hairline or minute differences between DNA, genes, and chromosomes. So this video is exactly meant to have that hairline difference to understand. Okay, the differences, the hairline differences between the DNA, genes, and chromosome. Okay, this is the very basic purpose of this DNA. So I have been getting this sort of question, okay, either in direct class or through mail. So I would like to make it as a video so that it will be helpful for everyone. Okay, so let us take a cell. Let us take a cell. So I have a very a basic diagram of a cell. This is going to be the cell. So what is going to be inside the cell? So inside the cell there is going to be a nucleus. So as I said, it's going to be either underdeveloped or well developed nucleus, depending upon the cell going to be an prokaryote or it is going to be an eukaryote. So inside this nucleus, what is that? Means, so there is going to be, so I will make it as I said, let me consider this as DNA. So inside the nucleus, let me consider there is going to be DNA present. Okay, and if I say DNA, so your automatically imagination goes to the structure of DNA. So which is a double-stranded helical structure. This is going to be the structure of DNA, which is the double-stranded helical, as proposed to by a scientist named Watson and Crick. So this everyone would be knowing about it, right? So then, what are chromosomes? What are genes? So all those things needs to be seen further. Right. So with this, so this is a double-stranded helical. So you know the meaning of helical. So it's some sort of a spiral structure. Okay. So we call it as an helical, and double-stranded means that there are going to be two strands. So in case I am going to separate this strand, okay, I'll have one strand like this, the other strand like this, which we normally notate it as 5 prime, 3 prime, and 3 prime, 5 prime. Okay. So the difference between these two structures. So here the both strands are associated together in case of this A, whereas in B there are two strands but they are separated. Okay, so separated structure, associated structure. So through hydrogen bond, all those things. Now, if I am going to consider this one strand alone, say 5 prime, 3 prime strand alone, I am going to consider. The question is, what are these strands made up of? Means all these, the entire DNA, all the organism, the entire DNA is made up of something called as N, which is nucleotide. Okay, the entire DNA is made up of nucleotide. So let me call it as N1, N2. So N3 it goes on till NN. So your 5 prime is going to start with N1 and it ends with NN. So this small n can have any value. It depends on the DNA. It might be 1000, it might be 10,000, it might be 20,000. Right? It depends on DNA. So you have from N1 to NN. Right? Now we are clear that DNA is basically made up of nucleotide. So what is nucleotide? This is a different subject, okay, different part of discussion. Now in this N1 to NN, let me take a particular stretch of sequence, say N1 to N150 or N1 to N200. Okay, so this particular stretch of sequence, some stretch of nucleotide sequence, okay, which is going to code for a single protein. Okay, this is going to code for a single protein. So you all know what, we are basically made up of protein. We are basically made up of protein, okay, in which the lipid, carbohydrate, everything is scattered here and there. So our basic biochemical nature is protein. So say in case if you have a consume a non-vegetarian food like fish, meat, you claim it to be a proteinaceous diet. Similarly, all living organisms are basically made up of protein, right? So let me consider this stretch. Say for example, it's going to be N1 to N150. Let me have it as 150 nucleotide. Okay, so this stretch of 150 nucleotide present in the DNA. From where I have taken this I have taken from DNA. So this diagram will make you the concept clear. So this stretch of nucleotide, okay, which is going to code for a single protein. So let that protein be whatever. So we have millions of protein. We humans have got millions of protein, of which any one particular protein, say insulin, the very common protein. Okay, insulin, right? So this stretch of nucleotide sequence that is going to code for this insulin protein will be referred as a gene. Here it is going to be insulin gene because it codes for insulin protein. So I'm going to have a definition for gene here. Stretch of nucleotide sequence, where it is present in DNA. It is present in DNA. What is it going to do? Can I take any stretch? No. The stretch should code for an individual protein, whatever be it. So you call that stretch of as gene. So here it is called as insulin gene. Likewise, if you take the entire DNA, it is made up of different genes. Say for example, if you have some 10,000 protein, then you should have 10,000 gene. So all these genes will be stacked, okay, and it forms a DNA. So I hope now you will be okay with, will be clear with the concept of gene. So how gene differs from DNA. So in order to summarize, I have to say gene all combined to form DNA. This is the concept. Like always you have a file, big file with some 10 folders. Each folder you allot it for a particular subject. Say first folder for physics, second folder for chemistry, third folder for botany, zoology. So the entire file here will be considered as a DNA. Each and every folder which is there inside the file will be the gene. So it is going to convey have the relevant information for that particular subject. Likewise, the gene is going to give the information, code the information for that particular protein. Again, what is this information? How protein information will be coded by gene is all a different discussion. Okay. So now you are okay with DNA. You are okay with gene. What gene is? The next question is what are chromosome? What are chromosome? So again, take this DNA. Double-stranded helical structure DNA. So this DNA, if you get it out of a cell and then measure, so average length of DNA will come around some 2 centimeter. Around our average, approximately around 2 centimeters. So if you are going to stretch it and measure around 2 centimeter the length. So you might be surprised that sir, see, we are not able to see our own cells. The reason is they are microscopic. So our eye doesn't have such a magnification. So we have to put our lens, microscopic lens on to view our cells, right? And inside cell there is going to be a nucleus, which is further a minute structure into which this centimeter length DNA has to be accommodated, right? It's a very tough task for the nature. Okay, so generally, if you see a very practical situation, that you have been provided a big bedspread and you have been asked to carry it to your home, okay, and you are planning to travel through flight or train, whatever be the mode. So what do? You have to accommodate a big bedspread, okay. I hope you all know what is a bed sheet or bedspread. So you have to put it in your suitcase. So what do? You generally compress it. You try to fold it, you try to compress it, okay. Then you will try to accommodate it in your suitcase. Then once you go back to your home, whenever there is a need, you again unwind it and then use it for your purpose. Similarly, the same solution, okay, the nature is going to provide here, that this, a bigger length DNA compared to the size of nucleus is going to be bigger. So this, DNA has to be accommodated inside a cell, okay, with a nucleus, okay, for which this has to undergo something called as compression, okay, something like compression, where this DNA gets associated with a protein which is called as histone. So this protein, consider this as a protein, it will be considered as histone, okay, and this structure, compressed structure or condensed structure, is what is referred as, okay, chromatid, and further they are called as chromosomes. Okay, further they are called as chromosome with all centromere, everything inside that. Okay, so DNA, the condensed structure of DNA is what is chromosome, just for the sake of accommodation. It has to get accommodated inside a very minute microscopic nucleus, for which DNA has to get condensed. Okay, so now it is quite clear that nucleotide all combine to form nucleotide will all combine to form gene, gene will all combine to form DNA, and DNA will get condensed to a structure called as chromosome. Okay, so if I say I have 23 chromosomes in my nucleus, I mean that I have 23 DNA. Or if I am going to say I have 46 chromosome in my nucleus, I mean that I have 46 DNA. And each DNA will have some sort of gene. And those genes, they are going to code for a particular protein. And each gene is in turn made up of nucleotides. Okay. So I hope now you have a very good understanding with the differences that exist between nucleotide, genes, DNA and chromosome, right? So hope this video has been helpful to you. Thank you so much.